Good morning, good morning, good morning. Same to you, thank you. Same to you. Good morning, good morning, good morning. So, everything all right? Audible, visual, everything clear? Same to you, dear children. Same to you, same to you. So, let's start. So, today we are just going to start with lysozo. Right? Up to Golgi complex we have discussed in last class. Right? It is also called what? It is also called suicidal bag. Got the idea? Right, lysosome. Now, if we talk about lysosome, how it is going to synthesize? Biogenesis. So what's happened? These are vesicles of Golgi body. Many vesicles of Golgi body. These are what basically? Golgian vesicle. And many Golgian vesicles fuse together. Many Golgian vesicles fuse together and make a large vesicular structure, making a somehow bigger vesicles. This structure is called what? Lysosome. So lysosomes arise from Golgi complex. Arise from Golgi complex. And if you want to say somehow more, you can say a term GERL. What is GERL stand for? Its full meaning is Golgi body which arise from ER forms lysosome. Means you got the biogenesis of Golgi body. So who is forming the Golgi body? ER forming the Golgi body. Who is forming the lysosome? Golgi body is forming the lysosome. Got the idea? Is it clear? Is it clear in the mind? Is it clear? Is it all right? Now we are moving ahead. So this is about how a new lysosome is forming. So it is a polymorphic cell organelle. It is a polymorphic cell organelle. Why it is polymorphic? It have a different, that is, it have different morphological structure in different physiological state. It have different structure in different physiological state. In different physiological state. I will tell you about it. I will tell about it. Got the idea? So what are present in the vesicles of lysosome? So inside the vesicles of lysosome, hydrolytic enzymes are present. Hydrolytic enzymes are present. Called, and these hydrolytic enzyme is called what? Acid hydrolases. Acid hydrolases. Some of the example of acid hydrolase means hydrolytic enzyme present in the lysosome is carbohydrate, protease, lipase, nuclease. Got the idea? So these are some of the example of which one? Hydrolytic enzymes present in the lysosome. Carbohydrate digest carbohydrate. Carbohydrate digest carbohydrate. Protease digest protein. Lipase digest lipid. And nuclease digest nucleic acid. What is this nucleic acid? DNA and RNA. So different type of. So I am just writing. I wrote here an example only. How many? 1, 2, 3, 4. But if you want more than 50 different acid hydrolases are present. Few example are this. Present in lysosome. Few example I already wrote in on board here. Present in lysosome. More than 50 different enzymes. Right. So optimum pH. Optimum pH means that pH at which it is showing the best activity. So optimum pH for working of acid hydrolase is 5. At 5 pH means 5 is what basically? Acidic pH. So at acidic pH, at 5 pH, these acid hydrolytic enzyme work in a best way. Got the idea? Is it clear in the mind? Is it all right? So now you just try to understand, this is a cell. I am just giving you an idea. This is a cell, and inside the cell, this is lysosome. This is lysosome, assume. And inside this lysosome, many acid hydrolases are present. These are so many different type of enzyme. These are acid hydrolases. So best pH is what? For working of this enzyme is 5. Now question arises, how it is maintaining the constant somehow 5 pH? So for maintaining the 5 pH inside the lysosome, here on the membrane a pump protein present. Who is present? Pump protein present. So when pH is increasing, this pump protein is pumping the H plus ion. H plus ion. What the idea? They are pumping the H plus ion. So when H plus, as more H plus ion, as less pH, you know na? pH means what? Power of hydrogen. Got the idea? As much H plus ion inside the cell, as much lower pH, acidic pH. So it is with the help of pump protein by the expenditure of ATP, they are maintaining which one? Acidic pH all the time. Getting the idea? When they are finding pH will increasing from 5, they start pumping proton. So when they start pumping proton, pH will decrease. Understanding the idea? And in such a way, lysosome most of the time maintaining which one? Acidic pH. Now I am just going to justify to you what I told. This is a which kind of organelle? Polymorphic cell organelles. Let me say once again it, so you will connect better. So I am just starting once again to say about lysosome. It will give you some better idea. Lysosome, it is also called which one? Suicidal bag of the cell. Why it is called, I will tell you, don't worry. So how the lysosome is forming? Many Golgi vesicle fuse together and forming the lysosome. Many Golgi vesicle fuse together and forming the lysosome. So in such a way, many Golgi vesicle fuse and forming a lysosome. And that is the reason I wrote lysosome arise from Golgi complex. And if you talk in somehow more detail, I wrote what? GERL. What is the full form of it? Golgi complex which arise from ER give rise to lysosome. Got the idea? So this is the meaning of this GERL, means it is helping you about the biogenesis. Later on I wrote about lysosome, it is a polymorphic cell organelle. Polymorphic cell organelle, getting the things or not? What is the meaning of it? This lysosome means it have different structure in different physiological state. I will explain just now this one. I'm just going to explain it, but let me finish it. So what I told, it is a polymorphic cell organelle. It have different structure in different physiological condition or different physiological state. Now I wrote inside the cycle of lysosome, which enzyme is present? Hydrolytic enzyme is present. This enzyme is also called acid hydrolase. Some of the example of acid hydrolases are carbohydrates, protease, lipase, nuclease. Got the idea? These are the some of the example of carbohydrate is going to digest carbohydrate. Protease is digesting protein, lipase is digesting lipid, nuclease is digesting DNA and RNA. Are you getting the idea? And now these are the some of the model example. What I wrote, more than 50 different acid hydrolases are present in the lysosome. And what are the optimum pH for working of these acid hydrolases is 5. How they are maintaining the 5 pH constantly? So suppose that if pH is increasing in lysosome, it becoming 5 to more. So in that circumstances, what happened? They are pumping the proton ion, and probably you studied about it. That much chemistry everyone know na. Even H plus ion concentration increase, pH decrease. Got the idea? Right? Is it? Of course, we will take care of everything when you came in the offline as well. Don't worry about it. Okay? So give your attention. So here you got the idea. Is it clear? Now I am going to tell you about function of lysosome, and then in last I will tell you how it is a polymorphic cell organelle. Function of lysosome. So first function I am writing intracellular digestion. Right, extracellular digestion. Third, you can say autolysis. Autolysis means where the cell is there, that cell is going to die. And due to this autolysis, it is called suicidal bag. Got the idea? Another function I am writing about the lysosome is Metamorphosis in frog. It have a role in metamorphosis. Metamorphosis in frog. Got the idea? So these are the some of the functions of lysosome I wrote here. Getting the things or not? Intracellular digestion, extracellular digestion, autolysis. Autolysis means self-destruction, self-breakdown. Getting the idea? Metamorphosis in frog, it have a role. Got the idea? Is it clear in the mind? Now I am going to tell you how these lysosome have different morphology in different physiological condition. It is important. So give your attention. This is a animal cell, assume. Here I am just symbolically drawing it is nucleus. And assume this is a primary lysosome. This is which lysosome? Primary lysosome. So I am telling polymorphic cell organelles means what? Lysosome I am writing. First let me write. Lysosome I am writing first primary lysosome. Then I am writing secondary lysosome. Secondary lysosome. Secondary lysosome I am writing of two type. Autophagosome. Another I am writing heterophagosome. You will understand everything, don't worry, just give your attention and connectivity. Third point I am writing is which one? Tertiary lysosome. Got the idea? It is also called residual body. A residual body. Got the idea? Now, what is this primary lysosome? What is this secondary lysosome? What is this tertiary lysosome? Let me write first, then I will draw everything through a diagram. So, primary lysosome. I am writing here. First form lysosome due to fusion of Golgian vesicle is called primary lysosome. Hydrolytic enzyme is present in inactive stage. In primary lysosome. Otherwise, you think, use your common sense, if in primary lysosome these enzymes are present in active stage, what happens? They are going to eat their own vesicle, they are going to eat the cells. So initially this enzyme is present in which one? In active stage. So in primary lysosome, you got the picture or not? So primary lysosome is the first form lysosome. Got the idea? First form lysosome due to fusion of Golgian vesicle, and in primary lysosome, hydrolytic enzymes are present in which stage? Inactive stage. Is it clear? Is it clear? Got the idea? Is it clear in the mind? So I wrote about the primary lysosome. Okay, done. So now we are going to tell you about some student asking previous slide. Don't worry, I will give you time to note down. Don't worry. So secondary lysosome you see first. Secondary lysosome, I wrote two type of secondary lysosome is there. One is autophagosome, another is heterophagosome. So first let me draw the secondary lysosome, then you will understand it better. Suppose that this is primary lysosome and suppose that this is a mitochondria. in the cell. But this mitochondria is old mitochondria. It is old mitochondria. Now not working. It is an old mitochondria. Now not working. Simply it is taking the space in the cell. Not performing any function. It become old. Old age mitochondria. Just try to understand. And suppose that here is food particle. Here food particle. So what cell did? Through endocytosis, they have taken the food particle. You can say they phagocyte the food particle. They capture the food particle like this. Getting the things or not? Assume they capture the food particle like that. Now food particle came inside the cell. Got the idea? Food particle came inside the cell and it is covered by a membrane, and this structure is called now which one? Food vacuole. Suppose that this is a amoeba cell and these amoeba cell capture the food from outside to inside. Food enter from outside to inside by the process of what? Endocytosis. Cell have taken, you can say phagocyte, the engulf, capture the food and taking the food inside. Now food are present inside a vacuole, that is called food vacuole. Inside the food vacuole, this blue color structure is what? Food. Now how the digestion of food occur? How the digestion of food occur? How intracellular digestion occur? Who will perform the digestion? Getting the things or not? So these lysosome play a role in the digestion. So these lysosome going to fuse with, this is lysosome, primary lysosome going to fuse with this food vacuole. And when it is going to fuse with the food vacuole, how the structure will appear? Let me draw. It is going to become a such kind of a structure. Inside here it is food is there. It is food particle is there. Got the idea? And in this area, who is there? Enzymes are there. Acid hydrolases are there. Now what happens? They will release this acid hydrolase on the food. Understanding the idea? And due to that reason, digestion of food occurs. So the structure what form here is called secondary lysosome. And when secondary lysosome formation occur, means when primary lysosome fuse with food vacuole, then their enzyme become active. Then their enzyme become active. Getting the things or not? So such a type of condition we are telling, this is out foreign body or inside the cell body? Food is a foreign body, which came from outside in the cell. So it is called heterophagosome. This is called heterophagosome. So when heterophagosome is what? When lysosome fuse with food vacuole. Food came from outside, it is outside source. It is making which one? Heterophagosome. You got the meaning of heterophagosome? It is a type of secondary lysosome. Now when the formation of secondary lysosome occur, this enzyme become active, this enzyme will release on the food, and who will digest? Food will digest, and after digestion of the food, simpler forms is going to absorb. Here absorption occur. So digested food now absorb inside the cell and they are going to utilize in the cell in the life process. Getting the idea or not? So in unicellular organism, digestion occur in such a way. In your body, digestion occur in which one? Alimentary canal, outside the cell. But in unicellular organism, digestion occur what? Inside the cell. And who have a role in this? Intracellular digestion. You got the meaning of intracellular digestion? I told now one function, intracellular digestion. How it is doing digestion inside the cell, you got the idea? Is it clear? Now this is a old mitochondria, unnecessary taking the space. What is the another possibility? Possibility number 2. This lysosome may fuse with old cell organelle. These lysosome either fuse with food vacuole or fuse with cell organelle. And when they are fusing with the cell organelle, a structure is forming. It is also a secondary lysosome. Here mitochondria, old mitochondria is there. So this is releasing enzyme. This is releasing enzyme, and who will digest? Old mitochondria will digest. So here it is also a secondary lysosome. Secondary lysosome. And this secondary lysosome is formed by fusion of which one? Primary lysosome plus cell organelle. It may be mitochondria, and then such kind of secondary lysosome is called autophagosome. And in secondary lysosome condition, who is active? Hydrolytic enzyme active. Enzyme become active. Hydrolytic enzyme become active. Did you got the idea? So are you very comfortable now in these two terminology? One is which one? This is called heterophagosome. I am putting line here. And this is called autophagosome. When cell organelle is going to fuse with lysosome for digestion, it is making autophagosome, and when food particle fuse, it is making which one? Heterophagosome. So there are two type of secondary lysosome, autophagosome and heterophagosome. Did you got the idea? Understand it? Clarity came in the mind, right? And after digestion, what happened? Digested part getting absorbed, and after absorption, non-digested part remain. So here a structure is forming. After digestion, after absorption, who is not digested is present here. Who is not digested is present here, and it is called tertiary lysosome. Let me write in short only. Tertiary lysosome. And it is also called residual body. A residual body, and it contains undigested component. And now this residual body come in contact with the plasma membrane and came out from the cell. Getting the things removed from the cell, expel out from the cell. Are you getting the idea? So how the intracellular digestion occur in the animal cell? You got the idea? Clarity came in the mind. How the intracellular digestion occur came, or should I say it once again? I just told how lysosome work for intracellular digestion. So these primary lysosome, secondary lysosome, tertiary lysosome are different physiological state. Different physiological state. Just a recap, please. Okay, listen it carefully. Listen it carefully. So I am just going to tell you about the lysosome, how it is performing the intracellular digestion. Got the idea? Tertiary lysosome not interact with the cell organelle. Only what is there? Primary lysosome fuse with the cell organelle, then forms. So let me tell you once again. I am just going to say it once again. Listen it carefully. Give your attention. So see, I am starting with primary lysosome. This is the primary lysosome. This is the cell organelle here. This primary lysosome is formed. Sir, can you please show the previous slide once? What is in previous slide? Let me check. Yeah, this is the previous slide. Okay, don't worry. You take a minute. I am not in hurry. Don't worry. So this is the previous slide. Is it possible to? This one you want? This one you want? Yeah, note it out. No problem. Who not did it, note it out. I'm giving you 1 minute. Done? Now come on the next slide. I am explaining it. So just give your attention. So this is the primary lysosome. It contains different hydrolytic enzyme. This lysosome is formed by fusion of many vesicle of Golgi complex. So in this primary lysosome, first form lysosome, all the enzyme are present in inactive stage. Got the idea? Now what happened? Just try to understand. Suppose that this is a amoeba cell. Suppose that this is a amoeba and here is the food. So what amoeba will do? It will capture the food and take the food inside by phagocytosis. Food came inside the cell. Now how this food will digest? Likewise I told here it is the food captured by the cell. And now question arises, food came inside the cell, present in the food vacuole, but how it will digest? So for digestion of this food is outside material. So this primary lysosome fuse with the food vacuole and making this structure. Their morphology is different from this now. Forming this structure, this is called secondary lysosome. This is called secondary lysosome. When secondary lysosome is forming, enzyme become active and these enzyme going to release on food particle and food particle get digest. And after digestion, what happened? Digested part get absorbed, and who is not absorbed is present as a which one? Residual body. Undigested food are present as a which one? Residual body. Getting the things? Here it is not digested. Here undigested material is there where no enzyme is working. So it is the residual body. Getting the things or not? So primary lysosome, secondary lysosome, tertiary lysosome. So this particular secondary lysosome is forming by fusion with the outside material, and then it be calling it what? Heterophagosome. We are telling it what? Heterophagosome. Are you getting the idea? Heterophagosome. Now these lysosome also cleaning the cell by digesting the unwanted cell organelle, by digesting the cell organelle who are not working. An example, this old mitochondria is not working. So what happens? These primary lysosome may fuse with this old mitochondria, and after fusing with these mitochondria, they are going to digest this mitochondria. So cell organelle are fusing with the lysosome. Self part is fusing. Hence it is called which one? Autophagosome. So after digestion, here also the same output will come, that is the residual body or tertiary lysosome. And then further, these tertiary lysosome and residual body, and by exocytosis, removing the undigested material from the cell. So this is the way digestion occur by the lysosome inside a cell. Intracellular digestion performed by the lysosome. Getting the idea or not? Is it clear? Is it clear? Right. Now one student asks, sir, then why lysosome is called suicidal bag? I am telling actually what happened. When your cell is in prolonged starvation, cell is hungry from a longer period. When who is hungry from a longer period? When cell is hungry from a longer period. What cell will do? These lysosome get rupture. Lysosome get rupture. Hydrolytic enzyme release in the cell and they digest all the cell organelle, every component of the cell, and cell will die after that. Hence the third function I told, autolysis. Check the function I wrote na, autolysis. Got the idea? Auto means self, lysis means breakdown. Got the idea? Death of cell occur. And that is the reason we call lysosome as suicidal bag. But autolysis, when it occur due to prolonged starvation. occur due to prolonged starvation. Are you getting the idea or not? Is it clear? Right? Done? So enzyme become active only when it come in contact of others. So when the enzyme came in contact with the other cell organelle, what happened? It become active. As long as it is present in primary lysosome, it is not active. Heterophagosome don't understand, sir. Let me tell you, auto or hetero, when lysosome digesting their own cell organelle, old cell organelle, unused cell organelle, the cell organelle who are not working, that is called autophagosome. When cell or lysosome digesting from outside material, got the idea? Suppose that food came inside the cell, and that food vacuole fused the life with the lysosome. It is forming which one? Heterophagosome. You got the idea? So first is primary lysosome, second is secondary lysosome. Secondary lysosome two type: autophagosome, heterophagosome. Autophagosome form when own cell organelle is fusing with the primary lysosome. Heterophagosome form when outside material which is coming from outside of the cell. An example, food. Getting the things? It is going to digest. Suppose that, getting the things, right? Lysis means breakdown. Got the idea? Residual body or tertiary lysosome, undigested material packed in a membranous structure, that is called what? Residual body. Is it clear? Is it all right? So you got the function. I am telling extracellular digestion. So I have given you justification of intracellular digestion. I told you about autolysis. Now I am telling about extracellular digestion. So where lysosome perform extracellular digestion? So in fungus, lysosome release hydrolytic enzyme on dead and decaying organic matter and digest it outside of body. Got the idea? So what, suppose that here it is some rotten vegetable are there, rotten vegetable organic matter are there, and on that rotten vegetable here fungus is growing. Suppose that here it is a fungal hyphae. Fungi will start. Suppose that this is a fungi which is growing. And what you understand better, I am drawing. Suppose that here a fungus is growing. Suppose that it is mushroom. Got the idea? So they are releasing enzyme, hydrolytic enzyme on food, and after digestion of food, these organic material outside, they are digesting, absorbing the food, absorbing the digested food. So digestion not occur inside the body, digestion occur outside of body. Are you getting the idea? Now I told it have a role in metamorphosis of frog. So you know that tail have what? Larva of the larval stage, tadpole larva have tail. Adult frog don't have tail. So who is digesting and consuming that tail? Lysosome is going to remove this tail. Got the idea? So tailless adult frog, tail is present in which one? Larval stage. So metamorphosis is what basically? Larval form to adult. Larval form to adult. So adult frog don't have tail, larval stage have tail. Who is playing a role in digestion of the tail? Lysosome. That got the idea? So that is the reason I told it have a role in, not it have only role, it have a role in metamorphosis of frog. Got the idea? So these are the some of the role of the lysosome I wrote. I hope you understand about it. And intracellular digestion, extracellular digestion, I think You got the entire picture of lysosome. Is it done? Can we move ahead? Lysosome over. So you know now what is primary lysosome, what is extracellular. Repeat. Okay. I am repeating extracellular. These are dead and decaying organic matter. This is mushroom, fungi. They are growing on dead and decaying matter. They are releasing the enzyme outside the body. They are digesting it in soil itself. And when digestion occurs, they are absorbing the digested food with the fungal hyphae. That we will talk about detail when we will tell about the fungi. Got the idea? Is it clear? So lysosome over. Now we are coming on the next cell organelle, and that is vacuole. So we are going to talk about three types of vacuole. One vacuole is large central vacuole. Another one is contractile vacuole. And another one is food vacuole. So large central vacuole present in plant cell. And it occupies around 90% of space Got the idea? It occupies around 90% of a space present in plant cell. So just try to understand with the help of a diagram about large central vacuole. Suppose that it is a plant cell. Just I'm making like this. Suppose that this is a plant cell. Got the idea? These are another cells are there. So this is a plant cell. So in this plant cell, you will find most of the area, around 90% of the area is occupied by vacuole. This is what basically central vacuole. The fluid present inside the vacuole is called cell sap. Cell sap. And the membrane of the vacuole is called tonoplast. Tonoplast. And fluid cell sap is also called tonoplasm. Got the idea? So what are present in the cell sap? What are the compositions of the cell sap? So in this vacuole, who is there? Excess water is there. Water, and what is fluid outside is called cytoplasm. I am just telling you differentiation between cytoplasm and which one? This outside fluid is called cytoplasm. So what is present inside the vacuole? It is excess water is present, sap is present, excretory product are present, and unused unwanted materials of the cell. Unused material. These are present inside the cell. So inside the vacuole, who is there? Water, sap, excretory product, or unwanted or unused material. It is working like a storehouse of the cell. Are you getting the idea? So what you kept in a store, what you are not using every day frequently. So the cell sap is hypertonic than cytoplasm. Cell sap is hypertonic than cytoplasm. Means it have more concentration. Hypertonic means high concentration of solute is there. Hypertonic means more solute concentration. Got the idea? So this is about large central vacuole. What is present inside the large central vacuole? Water is present, sap is present, excretory product is present, and unused and unwanted material is present. Membrane of vacuole is called tonoplast. How much area it is occupying? Around 90% of area of the cell it is occupying. Fluid present inside the vacuole is called cell sap. What are the concentration gradient of vacuolar sap? It is hypertonic over cytoplasm. So it is more concentrated than, and how the concentration is more here? There is a reason. So there is a pump protein once again here. Which protein? Pump protein. And these pump protein actively pumping the solute. Actively pumping the solute. This pump protein actively means by expenditure of ATP pumping the solute inside of vacuole, and due to that reason it have higher concentration. Did you got the idea? So this is about the large central vacuole. What is the job a vacuole is doing? Proton pump occur by lysosome, and here solute pumping occur by vacuole, and that is the reason inside hypertonic and outside hypotonic, less solute concentration. It is due to pumping of the solute inside the vacuole. Are you getting the idea? Clear in the mind? So this is about the large central vacuole. It is working like your storeroom. In your house, a storeroom job is what? What you are doing in the storeroom? What is the use of a storeroom in your house? You are storing anything which is in excess. Getting the things or not? Anything which is in excess and anything what you are not in use. Unnecessary thing. Got the idea? Is it clear? Okay. Now I am telling you about contractile vacuole. So contractile vacuole, it is present in an example amoeba. For your understanding, I am telling. This contractile vacuole is not present in every living organism. Amoeba is a single cell animalcules. So these contractile vacuole is responsible for perform osmoregulation. Perform osmoregulation. Now, what is the need of contractile vacuole? I am telling you will understand it better. Suppose that it is a freshwater animalcule, it is amoeba. Assume. I am just telling it is what? A amoeba. Now, in amoeba, it is present in, it is a freshwater amoeba, it is nucleus. And this amoeba is present in freshwater. So I am just putting here like what? Everywhere fresh water is there, outside. And inside is cytoplasm. Outside which water is there? Fresh water. Try to understand the problem. and inside cytoplasm is there. So cytoplasm is there. And cytoplasm is here hypertonic. Solution. And fresh water is hypotonic solution in compare to cytoplasm. Hypotonic solution. Water always move from the side of hypotonic solution to hypertonic solution. So due to that reason, what you will find, कि water continuously going inside the amoeba from the water. Now what happened? How much water it can hold? Excess water it can hold. Getting the things or not? So what happened? कि inside the amoeba there is a formation of vacuole occur, contractile vacuole, and where this excess of water is going to store. As water is coming, size of contractile vacuole become bigger and bigger. And a time came this contractile vacuole hit the cell membrane and removing the excess of water. So how it is helping the excess of water? You got the idea? What the excess of water coming, they are going to store in the contractile vacuole. Due to that reason, whose concentration not disturbed? Cytoplasm concentration not disturbed, and a time came, it hit with the plasma membrane and removing the excess of water. So excess of water coming, but with the help of contractile vacuole, they are expelling out these excess of water. First they are storing in this, then it come in contact with the plasma membrane, then they are removing the water. And due to that reason, they are protecting their own concentration. Whose concentration not disturbed? Cytoplasm concentration not disturbed. Otherwise, if this contractile vacuole is not there, concentration of cytoplasm get disturbed. Getting the things or not? So I am just telling you that it is working like human kidney. Human kidney. When we take more water, we pass more urine. Why excess of water removed by your body? Reason कि in your body a fixed amount of water is needed. If it is less than that, you are in problem. If it is more than that, you are also in problem. So these fixed water balance in your body is maintained by which one? Your kidney. So who is working like your kidney? Who is working like your kidney? Contractile vacuole in amoeba working like your kidney. Are you getting the idea or not? Is it clear? In your body, see now, when we are in our body, a fixed amount of water is needed. What the idea? Excess of water is expelled from the body. Same in the amoeba. Inside the cytoplasm, a fixed amount of water is required. If excess of water is coming, they will expel it. Understanding the idea? Is it clear? Right? Okay, ji. Thank you. Many of you not did the breakfast telling, okay, I'm sharing the QR code, don't worry. So you understood about the Golgi, which one? Lysosome and vacuole today or not? Fluid inside the vacuole is called sap. Thank you very much, dear children. Bye-bye. Have a good day to all of you. Bye.